Hello and welcome to another edition of Zog Science. Today we're going to talk about the evidence for evolution, go through some different examples, and yeah, it's gonna be cool. So, macroevolution. When we talk about evolution, there's really kind of two types. The first one is macroevolution. This is a large scale changes that take place over generally a very long period of time. We're talking on the time scale of hundreds of thousands to millions of years. And these are the large changes that create and eliminate species. So we start off with one type of bird here that then diverges into two separate species over a long period of time. And then we also have microevolution. So microevolution is a short time scale event that is basically kind of just changing the genotypes and phenotypes of a population, but they would still remain the same species. So we start off here with some white flowers and some pink flowers, and over time we would be left with only pink flowers. So evidence for macroevolution. We're talking about 5 different things: fossils, biochemistry, morphology, vestigial structures, and comparative embryology. So the fossil record. The fossil record just kind of shows us what has happened over time on Earth, what organisms were around, and how they have changed, because we get a glimpse at what they actually looked like. So fossils are the remains of ancient life, and when fossils are arranged according to their age, a progressive series of changes are seen. So we can line them up based on their age. How do we know how old they are? Okay, well, the first one is the law of superposition, and that basically states that a layer of rock is older than the layer above it. So if I've got one layer of rock on the bottom and then another layer on top, as long as there hasn't been any sort of seismic activity, earthquakes, fault slipping, anything like that, then we can assume that the one on the bottom is going to be older than the one on the top. And that's because over time, layers and layers of sediment build up on top of one another. Now this is mostly just works for sedimentary rocks, because those are the ones that again build up in layers. Metamorphic rocks and igneous rocks generally follow a slightly different trend of determining how old they are, but it does work for sedimentary rocks, and those are where you find fossils, because when igneous rocks or metamorphic rocks are formed, temperatures are too hot for fossils. And then we also have radioactive dating, and this is where we can use half-lives of radioactive isotopes in order to determine how old rocks are, and we can be very precise in terms of figuring out exactly how old a rock or fossil is. So here is some horse fossils. Okay, so we can see we've got Eocene rock, which is an epoch. It's fairly old, 60 million years ago, and you can see the how the differences in the bone structure has changed over time, right? So as we get younger and younger rocks, more recent rocks, the bones that we find of horses, those have very different structures, and we can see that they originally started off with these kind of 5 points, right? 1, 2, 3, 4, 5. And then it has slowly, 2, 3, 4, 5. The 5th one is actually up here near the knee, and they have slowly changed over time. Same thing with the molars, right? The teeth, those have changed over time. Here is the evolution of elephants, right? So we can kind of see how we have different structures. Again, these are all based off of fossil evidence that has been found. And here is the cat family. So we've got, I believe that we've got all kinds of different trees. They broke off here before this. Here's your saber-toothed tigers, as they're called down here, and your different panthers. These are your things like tigers, lions, etc. Felis, this is your domestic cats. So we can kind of see how they have broken off of one another and evolution has occurred. Again, we look at the structural differences in the fossils, and we can kind of see the changes that have occurred over time. So there are gaps in the fossil record, and they do shrink as new finds are made. But just because there's a gap does not necessarily mean that that's a weakness of the theory of evolution. It just means that we haven't found the fossil yet, and that we don't necessarily understand how some species have evolved. but the general picture is still there. So it's not like we can throw out evolution just because we don't have the one fossil that is exactly in between any two species or any two fossil records. We also have comparative biochemistry and molecular biology. So what we do with that is that we compare the base sequences, so the DNA base sequences in genes. We compare amino acid sequences in proteins. So in this case, we're looking at the DNA nucleotide of the cytochrome oxidase gene. That was the COX gene that we looked at when we did the bioinformatics lab. It is a protein that is found in the mitochondria. It's one of the last ones in the electron transport chain, which we mentioned was a part of the process of aerobic respiration. So what we can see is that when we compare the DNA to one another, what we're looking for is the differences. Okay, so the majority of this gene, which is, as you might recall, is maybe 500 or so base pairs long, right? The majority of those letters are the same. However, there are differences, and so what we can do is we can try to rank them, the different organisms, based on how many differences there are in the genes, and we can try to come up with an idea of which ones are more closely related to one another, right? So humans and pigs are both mammals, so it makes sense that they would have fewer differences than, say, humans and ducks, right? Ducks and snakes are both closely related. Birds are actually more closely related to reptiles than they are to mammals. So we see that there are very few differences there. And so on and so forth, we can kind of split them apart, see that yeast are very, very different from the rest of these, which again makes good sense because they are fungus, whereas all these are other ones are animals. So all organisms from bacteria to humans have many of the same biochemical details. What that means is that they have a lot of the same proteins. And so the genetic code for the protein coding genes is nearly universal. All organisms use ATP, right? So every single organism in the entire world uses ATP as an important energy carrier. Why would that be unless they all kind of started off from a similar place? Also, the plasma membrane of all organisms is a phospholipid bilayer, whether or not that is a eukaryotic or a prokaryotic organism. So the number of amino acid differences between hemoglobin and other organisms. So this is a different protein, hemoglobin, right? We're comparing the different organisms. Again, we see that the differences from humans, right, is getting larger and larger. And again, as they do so, they become less and less similar, right? So these are all mammals, less differences, and we got birds, frogs, amphibians, and then lamprey, which is a type of fish. You have more and more differences accumulating the less closely related they are to one another. We can also use this to determine kind of the primates and determine who is more closely related here. So if human and chimps are more closely related, aka they have a common ancestor more recently, then we would expect the DNA sequences of humans and chimps to be more similar as well. So if we take a look, right, humans and chimpanzees have fewer differences in the mitochondrial DNA. They have fewer differences in the testes-specific protein on the Y chromosome. And then they have fewer differences in terms of the globin gene, so that is the hemoglobin gene. What they mean by non-coding is the areas of the gene that are that aren't involved in actually producing the protein, but more so the regulatory sites, the places that help regulate the gene expression. Another evidence for evolution is comparative morphology. Morphology just kind of means the structure of the organism. So if we look here, we can see that whales have 5 fingers in their fins. Cats have 5 fingers on their toes. Bats have actually 5 fingers in their wings as well. And then a gorilla has 5 fingers in its hand. Notice that we kind of have these bones up here that are very similar, and then the ones up at the top. So homologous structures, that's what those were called, are parts of different organisms that are often quite dissimilar that develop from the same ancestral part. So all vertebrates, which are those that have a backbone, contain a similar pattern of organs, which suggests that they'd be related to one another. And the forelimbs of all mammals contain the same pattern of bones, although the bones now carry out a variety of different functions. So the function of a whale's fin is to swim. Function of a cat's paw is to walk. Bat's wings fly. A gorilla's hands grasp things, right? They hold on to things. So they all have different functions, but they all have very similar structures to one another. And homologous structures teaches an important lesson about evolution, which is that evolution works primarily by modifying pre-existing structures. It's not about taking, building a completely new structure from nowhere. You have to start with something, and then it slowly gets modified over time until it has a different function. So even when two species function in completely different ways, they use those same structures to carry that out. So what happened originally is that we had an ancient lobed-finned fish that eventually started to walk onto land, and over time, the bones that were inside were modified or changed to survive in different habitats, different environments, and so we get different bones that have come out with that for different people, different structure, not different people, but different animals, different structures, different functions. So all tetrapods, which are four-legged organisms, have limbs with 5 digits, at least at some stage in development, whether or not that's in the embryo or later on. And certain tetrapods lose some of these digits during development, as in the birdwing, which is right here. So the birds actually have 5 when they're in the egg, but then they lose 2 of them and kind of go down to 3. But it doesn't really make sense as to why they would have it in the embryo if it wasn't something that used to be in one of their ancestors, something that they actually had in their wing, and right now it's kind of being lost over time. But we do see that all of these organisms, they all have a humerus, right? That large bone up at the top in your upper forearm. They all have a radius and an ulna, right? So that allows the rotation. Okay, that allows the rotation of your arms, and then they all have your metacarpals, which are your bones in your hands. And again, the most plausible explanation as to why they only have the 3 is that they are a genetic artifact. Here's the opposable thumbs, right? So chimpanzees, humans, gorillas all have opposable thumbs. We can also have analogous structures. Analogous structures have a similar form and function, but are structurally quite different. So think about like bird's wing and an insect's wing. Both of them have the same function, and they kind of look similar in terms of they're both used for flying. However, they're completely different structurally, right? This is made up of just protein and it doesn't have any bones inside of it, whereas the bird's wings has feathers and bones. And that's based on the fact that both of these structures are used to do similar things, right? They're both used to fly. They just have evolved from different things. So it's a similar environment that has forced this evolution, or not say forced, but led to this evolution, but it just has come from a different place. We also have vestigial structures. So many organisms contain structures that no longer function, but resemble functional structures of other organisms. So what this suggests is that the structures are inherited from a common ancestor. For example, humans possess a complete web of muscles for wiggling their ears. So if you might know some people who can actually wiggle their ears. That's because they have muscles that are specifically attached to it. You think about your dog or coyote or cat or whatever. They have the ability to actually rotate their ears, right? That's because we evolved from a common ancestor that they had many millions of years ago. And so our muscles are still there. We've kind of lost the majority of our control over them. Humans also have an appendix. Some of you might have had your appendix removed. It is used or it was used in the past to help digest certain foods, mostly plants, but we no longer really need that at this point. And so it has kind of withered and is essentially on the way out. If we were to look at humans in several million years, we might not find an appendix in any of them. And then whales and snakes contain pelvic bones, just as humans do, but they have no function, right? Because snakes, they slither on the ground, and whales swim, so they don't need a pelvis. So here is that pelvic bone, right? So there's a salamander with his pelvis. There's a baleen whale. You can see that it still has a pelvic bone right here. And then here's the snake. Again, it's got a pelvis right here. So just kind of some of the vestigial structures that we humans have. One of the main ones is the coccyx, which is the tail, right? So if you've ever heard somebody falling on their tailbone or breaking their tailbone, there's literally a bone that it protrudes down below the pelvis that originally was a tail that we had. So here is a child that was born with a much longer tail. So there are some people that are born with tails. Generally, they are surgically removed at birth. We can also take a look at embryology, right? So pharyngeal slits are kind of the precursors to gills. And what we can see is that in the embryos, right, all these embryos look very similar to one another, and all of them have pharyngeal slits. So again, that is evidence that all of these organisms evolved from a common ancestor because they all have very similar embryos. So during development, organisms may exhibit characteristics of other organisms. That's kind of something that we just kind of see. And these suggest that the organisms are related to one another. So early in development, embryos possess gill slits like those in fish. Those are those pharyngeal slits. And later in development, the embryos develop a tail, right? And human fetuses have a fine fur or lanugo during the 5th month of development, which is something that a lot of our mammal relatives also have, that fine fur. So if we were to take a look at all of these different embryos, right, we would all want to say that, hey, these are fairly related to one another, right? So let's kind of take a look. Which one do you think is the human? Make a choice. All right, so we can see kind of as development progresses that they all started off very similar, but we're starting to see some differences, right? These two over here have become very different from the rest, but we've kind of got, maybe a kind of a grouping over here, but these 4 over here still look very similar. So here's our human. We've got, I believe, a cat, a lamb, a pig, a bird, and then a turtle, right? So they all look very similar to begin with. And again, this suggests that there was a common ancestor at some point, and then they begin to diverge as they develop. That was a cow. That's what it was. So there's that lanugo, that fur that you get so this would be a preemie baby that was born a little early. And microevolution, okay? So microevolution, some of the evidence, evolution of pesticide or antibiotic resistance in response to selection. So what we have here is we have a bunch of pests, okay? One of them has a chromosomal mutation that allows them to resist the insecticide, right? An insecticide is used, killing off all of these other ones that do not have that resistance. The survivor lives, right? And then it is all of its offspring are going to have that particular trait. So that is evolution, right? The new population has a trait that not necessarily everyone in the population had before it. We call that microevolution. We also see microevolution occurring in HIV drugs, right? So HIV drugs a lot of times have to be completely renewed all the time because the HIV virus has lots and lots of mutations. So we see patients that a drug is effective briefly, but then it is all of a sudden not effective anymore because that virus has evolved to be resistant to it. They talk about lots of drugs having this problem, right? So there's an antibiotic issue in the United States where antibiotics are used. They kill off all the bacteria in your body that are causing a disease, except for maybe the 1 or 2 that have resistance to it. And then those 1 or 2 bacteria begin to multiply. They have offspring. All of their offspring now again have this antibiotic resistance. So this is vancomycin, and it was used in the 90s. And we can see how the percent of those that it had no effect of was rising dramatically just in a few years, right? This is just 8 years right here that it kind of shot up, and the antibiotic resistance was increasing. So again, just kind of another way to look at this is we've got our bacteria, right? We've got one that's resistant. The antibiotics are used. Most of the normal bacteria die. Again, the resistant ones begin to multiply, and you're left with bacteria that are only resistant. All right? And that's all we got for you today. Thanks a bunch for watching, and I'll see you next time.